First of all, thank you everyone for coming and giving an opportunity to present our work on a new data set that we've developed within our lab. It's a pan-cancer open data set for nuclei classification and segmentation, instance-based, and it's been a equal contribution work with my collaborator Navid and consulting pathologists that work with us and my supervisor Nasir Rajput. So first, a brief announcement. Later on today, we will be doing a tutorial on machine learning for pathologists because I really believe we need to close that gap in terms of knowledge and asymmetric information that we have in terms of understanding the algorithms that we use. And that way, we could definitely speed up the development of new methods and discovery of new biomarkers, which could significantly contribute to the whole community. And we also are curious to gauge the interest on how much people are willing to learn about machine learning. So if you could complete that survey later on when you have free time, we would really appreciate. So why PanNuke? Why, you might wonder, why do you need a new data set for nuclei classification and segmentation? Well, there are several reasons why. First reasons why we decided to do it are clinical, and particularly for biomarker discovery and other sort of downstream analysis. But there are also technical motivations that I'm going to touch upon. So in terms of clinical motivation, obviously we can do all sorts of analysis in terms of cancer grading and biomarker discovery. But what I would particularly like to touch upon is tissue phenotyping, which is a work done in our lab, where they classified different nuclei within the visual field and then construct a graph. And then on that graph, they perform, what's that? It doesn't go back? Oh, okay. So yes, you detect different nuclei, you classify them, and then you construct a graph based on the connections between the nuclei. Based on that, there seems to be an issue with an image in the slides. Okay, you construct the graph. Based on that graph, you are then able to do community detection on the whole slide image, and based on those communities, you are able to identify different tissue subtypes within the whole slide image. And that allows you to provide essentially unsupervised annotation for the whole slide image and discover all of the tissue subtypes within the specific tissue. Now that study was performed only on colon tissue, while data set like PanNuke would allow it to do it on multiple different tissues. And unfortunately, we don't have the images for that. So therefore, we go on to technical motivation. So in terms of technical motivation, it's really about size and scope. So these are an example of several available data sets for nuclei classification and detection. So Kumar dataset has been part of Monoseg challenge for nuclei segmentation, and it only consisted of 30 visual fields. The dataset that was released as from our lab for nuclei detection in colon only consisted of 100 visual fields, and recent challenge on nuclear cellularity scoring in breast consisted of 155 visual fields. And our data sets in total consist of 455 visual fields, and the numbers keep growing. Now, in terms of the scope, the Kumar data set provides only 7 different tissue types. Then the data set released for nuclear detection in colon has obviously just colon, and the recent nuclear cellularity challenge consists only of breast images. Now, PanNuke, hence the name of the nuke, consists of 19 different tissues that would allow to essentially, if you use community detection, semi-automatically annotate almost all of TCGA. And also important note here is label sparsity. Most of the data sets that existed in the past, oh, there is a TV here, I didn't know. So most of the data sets that were released in the past consisted only of, either didn't have any annotations in terms of nucleotypes, so for example, Kumar, or colon dataset consisted only of 4 nucleotypes, and these were not exhaustive. And same with the cellularity challenge in breast PathQ. And this label sparsity does not allow one to train end-to-end encoder-decoder-based models. So you still have to restrict yourself to patch-based classification models. While PanNuke, because it was constructed semi-automatically, all of the nuclei within the visual fields are labeled, and that allows to train end-to-end models. Another important point is sampling bias, which has been briefly touched earlier in the talks today when people were discussing how big data sets are sufficient for training deep learning models. And it's really not just about the size of the data set, it's also about the diversity of the data set and how that data set is representative of the whole population that the algorithm might face in the real world. So here's an illustration. Let's suppose that we have multiple different colors that these represent the diversity within the population of visual fields that an algorithm might face in the real world. And let's say that, for example, black dot corresponds to an artifact and a visual field with nuclei, but also with an artifact. And usually the data sets that we see in public, they will try to exclude the artifacts and therefore train our models on cherry-picked samples. Well, that makes our models vulnerable when we try to deploy them in practice to these artifacts. And this might affect downstream analysis significantly. Oh, wow. So we've conducted an experiment of sampling bias in practice, and I apologize for graphics screwed up. So these are 4 visual fields from the Kumar dataset, on which we trained our detection model. And the specifics of these Kumar visual fields is that they are really, really beautiful visual fields. There is nothing going wrong in them. There is just beautiful nuclei there. Obviously, you can't see in this picture. And these are what we essentially mean by cherry-picked visual fields. They have lack of artifact diversity, which are abundant in clinical routine, and they most likely are restricted to the same magnification and scanner type. So what we did, we trained a nuclei detection model on these visual fields, and then we applied this detector to unseen visual fields randomly sampled from whole slide images. So what we find then is each dot on these visual fields is a detection by the model. And the yellow one is what corresponds to an actual nucleus, and the red ones, which should not be detected by our model because this is an artifact and it should not contribute to the downstream analysis. So this is what happens when we use currently available public data sets. And this might not necessarily be good. Again, screwed up image. So this has been shown in a clinical study where they took an algorithm, deep learning algorithm trained on academic data and apply it in clinical setting. And what they've seen is the significant underperformance of the deep learning algorithm in a clinical setting. And this is what we should be really careful about and really think about sampling bias when we are creating new data sets for any of the studies that we're conducting with machine learning models. So what is PanNuke? So it is a semi-automatically created nuclei instance classification dataset and segmentation dataset, which consists of 455 visual fields, of which 312 were sampled from different whole slide images at different magnifications and from different data centers. It has nuclei labels for 19 different tissue types, and we continue to grow the size of this dataset. So how did we do it? It's essentially an active learning based approach which allows a model and a pathologist or any kind of person who develops the experiment to work together. We train the model on available data sets, and then we use the uncertainty of the model to query which sample we should annotate next. There were 3 parts to our approach, each consisting of training the model on labeled data, then model labeling unlabeled data, and then selecting this unlabeled data and validating and annotating by domain experts. So the first part consisted of 3 separate ensembles, which is just multiple models for a specific task. These were detection models, segmentation models, and classification models, which we trained on public and internal data sets that we have available. So detection and segmentation models were encoder-decoder type networks, and classification models for classifying the nuclei were patch-based classification networks, mainly in ResNet, if anyone is interested. So we trained this on the data sets that I've talked about earlier, such as Kumar and the colon data set that we have internally, as well as the breast data set. And then we label the randomly sampled visual fields and then select those based on uncertainty to be labeled by our pathologists. For classification, these are the details of the data sets that we started with. So we labeled some of the visual fields from monosec. We've also had an internal colon data set, as well as the available breast data set from the recent challenge. And also we used nuclei attributes data set, where we've picked the category of no nuclei from there, which we found very useful to detect those false positives produced by detectors trained on cherry-picked samples. In part 2, once we've completed part 1 of several iterations of part 1, we've created a new whole visual field detection and classification model, which was not per patch classification model, but encoder-decoder type network. which allowed us to incorporate context. And this is something that we found when speaking to pathologists, that they do take a look quite a bit at the surrounding nuclei whenever they're making a decision about the nuclei that they're looking at. And we've performed exactly the same steps. We would train the model on the data that we have labeled already. Then we would label unlabeled data once again, and then select those for annotation. And we would do several iterations. Okay, so imaging doesn't work again. So where could you get this data set? You could either scan this QR code, or you could get it from the link which is visible right there. And we are releasing, as part of this data set, segmentation models, classification labels, detection labels, and also trained models, as well as the statistics about the model performance, which is in preparation, and also data exploratory statistics. And also these slides and references can be found on the link as well. Yep, that's about it. Thank you.
Any questions? You said that you, if I understood correctly, you try to avoid the areas of artifact, right, when you create this ground truth?
No, it's the opposite. We try to have artifacts.
Include them. Okay.
We try to mimic the real environment that the algorithm might work in as much as possible.
So if you can explain the reasoning for that, just the importance of it.
So the importance of it is the concept of sampling bias. We want our data set to be representative of the population, of the population of all the possible examples that represent that we might face in the pathology imaging.
In the SPIE challenge dataset, if you downloaded the whole slide images, you would have found some actual nuclei, point-based classifications for TILs, normal, and malignant.
Yes, this is exactly what we've used. This is what I mean when I've mentioned SPIE.
So you had 0 for the normal cells in that spreadsheet. So there were lots of normal epithelial cells.
So from there, we mainly sourced malignant.
Oh, okay.
Because we had quite a few already for normal ones.
Jason?
You mentioned more data coming in. So is there an end in sight? Will you have sampled everything, or how does this go forward? And what are your thoughts
this is why I mentioned that in preparation is the fully working model. And what we essentially want to do is a cross-validation over the whole data set and determine what is enough for the model to be working on all 19 different tissue types.
But that means the model will work on the data set, right?
Yeah.
Yeah.
And so, I mean, it's PanNuke. So
yeah.
The view is to have a model that works on all data.
Yeah.
I guess
yeah.
How do you see getting there? That's really what the question is.
So getting there is the point at which we stop is when we're satisfied of having enough data that we can learn good enough model to classify nuclei in any of these tissues sufficiently. So at the moment we have around 20 visual fields for each of the tissue types, but this is not sufficient, what we find, because some of the nuclei, for example, I think prostate and breast, they might look similar but correspond to a completely different class. And this is the challenge that we face.
Let's thank our speaker one more time.
Thank you.